Dr. Borup here to talk to you about enzyme activity. This is Lab 3 for Anatomy 36. We'll begin with an overview of the digestive system. The purpose of this lab is actually not to study the digestive system so much as it is to study the effects of concentration and pH and other factors, temperature, and so forth on enzyme activity. Nevertheless, digestive enzymes make a great tool for studying enzyme activity. They're very easily obtained and are fairly cheap, and so we can get them on the market without too much problem. We haven't had a chance to talk about the digestive system. We'll actually go into we'll have a full lecture period devoted to the digestive system, but that's not until later in the semester toward the end. And so I want to begin with a quick overview of the digestive system. On the left, you see an image illustrating the structures that are devoted to the digestive system. They should all be familiar to you from your anatomy class. The mouth, of course, which is going to begin the process of digestion through mechanical digestion. This would be mastication or chewing. The mouth also contains enzymes in the saliva, which are going to participate in what is known as chemical digestions, and these are chemical reactions that occur. They're catalyzed by various different enzymes that are produced by the digestive system. Included with the digestive system are some accessory structures, such as the liver, the gallbladder, and you can't see it very well, but tucked in here is your pancreas. And those structures are going to participate by producing enzymes and other chemicals that are going to help with the digestive process. In addition to chewing, mechanical digestion includes the mixing in the stomach. Your stomach actually contains 3 different layers of muscle, and these muscles will mix and churn and kind of squish up the food that is contained within the stomach, mixing it with the chemicals, namely hydrochloric acid and an enzyme called pepsin. And then we, of course, have peristalsis, which propels the food forward. This is the contraction, the circular contraction of tubes. Contraction will start, for example, in the esophagus, in the deglutition reflex. That's the fancy word for swallowing. We begin as a bolus of food, moves from the mouth into the esophagus. It is propelled downward by systematic contractions of the muscles in the esophagus, contracting here, which will push the bolus of food forward to here, for example, and then we'll get another contraction which will push the bolus of food forward even further, and so on and so forth. This is peristalsis. We'll look at this mechanism in more detail later in the semester. Here's a closer look at those accessory structures that I mentioned. This includes the liver and the gallbladder and the pancreas. The liver produces something called bile salts. Bile salts are important for the digestion of fat. They are not enzymes. We will look at those in more detail in a few slides, and we'll also discuss them again later in the semester. Bile salts are synthesized by the liver. They are stored in the gallbladder, this little structure that you can see right here. Let's get a different color that actually shows up. Let's try this color. Not much luck. Okay. We'll try just this. There we go. There we go. There's my gallbladder. It's tucked in underneath the liver, and it contains, let me show you these ducts. I'm going to outline this. Okay. Here we have our bile duct right here. So here's the common bile duct. Okay. And it's going to empty into the duodenum, which is the beginning portion of the intestines, of the small intestines specifically. So this region right here. Okay. Bile will dump directly into that area. It is stored temporarily in the gallbladder, as I mentioned, but you can remove the gallbladder, and this is actually a somewhat common surgery. If you end up with gallstones, these are hardened deposits made of bile salts that crystallize. The gallbladder can become quite painful in those situations, can cause a number of digestive issues, and sometimes the decision is made to completely remove it. There's sometimes consequences to that, some sensitivities to high levels of fat and so forth, but you still have bile salts being emptied into the intestines due to production in the liver. So remember, the gallbladder is just for storage. The liver is actually producing it. The pancreas is a very important and interesting organ. It has endocrine function. Endocrine. Crin means secretion, endo means inside. And then it also has exocrine functions. Crin meaning secretion and exo means outside. What on earth does that mean? Well, the endosecretions are your insulin and your glucagon hormones, which are secreted into the bloodstream. And we'll have a chance to review the control of insulin and glucagon when we talk about the endocrine system. But exo means to be secreted to outside. And here I want to remind you that the lumen of your digestive system is the external environment. That scribble says external. This can be a little hard to read there. The external environment. And so the pancreas, what it does is it dumps a bunch of secretions into this pancreatic duct. And then if you follow that duct down, okay, yellow was not a good choice, let's try red. Follow the duct down, you'll notice that it's going to join into with the bile duct and dump its secretions into the duodenum of the digestive system. Notice that there's a separate, more minor duct right there, and so can also dump that in separately. The enzymes produced by the pancreas, we're going to go over these different secretions in more detail, and you are going to want to know that a significant portion of the secretions that go into the digestive system, specifically the intestines, come from the pancreas. Speaking of secretions, let's make sure we define that term for you. What we have here on the left is a very beautiful picture of the lumen. Let me make sure we get that written for you. So this is the lumen here of the digestive system. This is a segment of intestine. You can see the beautiful pseudostratified epithelial here. Let me identify a goblet cell for you. I want a different color. These beautiful goblet cells are specialized epithelial tissue that are there to secrete mucus. And then if you look right here, okay, you can see how it looks a little fuzzy there. That is what is known as the brush border, and it's created by villi, which are little finger projections. So if you were to zoom in on the apical membrane of the cells in the digestive system in the intestines, you would see these finger-like projections, like so. And these are the villi. And then if you were to zoom in on a segment of those and look at just one cell, okay, what you would actually see is finger-like projections on the single cell, and those are called the microvilli, or villi, and they are also referred to as the brush border. You'll want to review your anatomy if you're not really comfortable with that, or hit me up for a little bit more in detailed tutorial. Here we have the basement membrane. Let's pick a different color. We're going to pick green. Green sounds good. Basement membrane. And if you look right here, this is a capillary. And that's pretty important to understand, that we have capillaries running beneath the lamina propria, and that those capillaries are going to be what's picking up the nutrients from the small intestines. Secretion involves the transport of substances from the lumen from the cell into the lumen. And these substances might come from the blood. If that's the case, they're going to go from the blood, so from these capillaries into these cells, and from the cells into the intestines, into the lumen of the intestines. So that's one form of secretion. Another form is not shown here, but we would see it with the endocrine system, where you would have endocrine cells like so, and perhaps they would be surrounding a capillary, or at least closely associated with a capillary, and in that case they would be secreting their substances into the capillary. So both are secretion by definition, either moving substances from the cells or from the bloodstream into the lumina of the intestines, or moving substances from an endocrine gland into the bloodstream, or from any type of cell into the bloodstream. Other cells can secrete substances too, like neurons, for example, will secrete chemicals into the synapse. Transportation of these molecules, if they are water soluble, then they're going to be transported with carrier proteins, either through facilitated diffusion or active transport, depending on what the concentration differences are in the cell. I'll give you an example. Concentration of sodium, actually let's use hydrogen ion instead. Concentration of hydrogen ion in the stomach is going to be very, very high. And yet we're going to want to secrete more hydrogen ion into the stomach, and so we would have to use, anytime we're going against a concentration gradient, we're going to be using an active transporter. And so there are indeed what are called protein pumps in the stomach that are going to be responsible for that. Now in the intestines, the concentration of hydrogen ion are actually quite low. We'll get into that again in more detail when we hit the digestive system at the end of the semester, so don't stress too much about the mechanism right now, but we will be looking at the molecular mechanisms later. Transport also includes those molecules that are lipophilic. Those are going to be transported by diffusion. Remember that they are simply going to cross the cell membrane without the need of any kind of carrier protein. We don't need an ion channel, we don't need a transporter. They can just go right through as long as they're small. Larger molecules have a difficult time, and so molecules such as triglycerides actually need to be broken up to small fatty acids and glycerol before they can get through the membrane due to size. And then last but not least, we can also transport paracellularly. This is a less common way of transport in the intestines, but it does happen. We could perhaps, for example, transport water into the lumen through paracellular transport, but as I mentioned, that's less common because we tend to want to regulate that, and so we're going to have gap junctions in place to prevent that process. And then transcellularly, you of course know that we're going to have protein transporters at the basolateral membrane, so we'll have transporters here which are going to transport some substances into the cell. We'll have a different set of transporters out here which will transport substances out of the cell. So you have different transporters in the basolateral membrane and in the apical membrane, and so that allows for molecules to be transported through the cells and into the lumen of the digestive system. And then last but certainly not least is exocytosis. This is less common in the digestive system, although it is used. It is more common in cells such as neurons and endocrine cells. They'll actually use exocytosis as a method of secretion, and so you'll hear me talk about later in the semester secretory vesicles. What I really mean is we're using exocytosis to secrete large substances into the interstitial fluid, and from the interstitial fluid into the bloodstream. Absorption essentially is the opposite of secretion, in that instead of pushing substances into the lumen, we're actually pulling substances out of the lumen, of the intestines in this case. And so we are transferring molecules through the apical membrane. So here with transporters found in the apical membrane, and we transport them generally transcellular once again, and then from the basolateral membrane we'll transport those molecules into the bloodstream. So a molecule will simply be transported like so until it gets into the capillary there. From the capillary to be transported to the rest of the body. Transcellular is much more common, of course, than paracellular, but again, some paracellular transport occurs. For example, in the large intestines, we get some transport of water reabsorption that way, or water absorption that way. You'll hear from time to time me use the term reabsorption. As long as my mouth isn't talking faster than my brain, reabsorption is typically used for things like the renal system, and the reason is it recognizes the fact that a substance must first be absorbed once in the intestines, then secreted or filtered in the case of the renal system, and then reabsorbed if we pull it back into the bloodstream. And so it's just a distinction between the number of times it's been absorbed. First absorption is in the intestines, second would be from the renal tubules, for example. Again, we can use endocytosis. That should not say endocytosis. Yes, endocytosis. Yes, it should say endocytosis because we are talking about absorption. Exocytosis is about secretion. And just like with exocytosis, when we start talking about those lipophilic molecules, those can actually get through the apical membrane through simple diffusion as long as they are small. If they're larger molecules, they actually need to be brought in by endocytosis. Then excretion, that's fairly straightforward. This is the removal of a substance from the body completely through urination, defecation, or exhalation. Don't forget that the exhalation of carbon dioxide is how we actually secrete CO2. Or not secrete, excrete CO2. Of course, through the digestive system, those excretions tend to be solid waste materials, including fiber and any undigested food particles. And that material is transformed to remove water, as much water as possible, so that by the time we actually create a solid waste, it is 1% of the total water that was in the digestive system at any given time. Let me double check that number. We actually have it down here. So this actually shows you that nice fluid volume, some kind of wow factor here. In that, we usually take in about 200 milliliters of fluid through both our food and our drink. This is a fun fact. You produce 1.5 liters of spit every day. Kind of crazy. 1.5 liters of gastric secretions. Gastric means stomach. 1 liter of bile salt. Another liter of pancreatic juices. This includes water plus all the enzymes and buffers and so forth. 2 liters of intestinal secretions. Grand total, you're dumping, okay, the 72 milliliters of secretions plus the 2,000 milliliters of whatever you're getting from your food and your drink. That would be a lot of fluid going through you. 900, 200 milliliters, okay? That would be a lot of fluid to go tearing through your digestive system and being excreted, and you would become dehydrated very, very quickly if we didn't reclaim that. Fortunately, we reclaim most of it, leaving only about 150 milliliters of fluid lost in the digestive system from the feces and the feces. Okay, pretty crazy how much water is going in and out of this digestive system. Now let's look at the secretions. We'll start with the mouth. These are important secretions in the mouth. Some are more important than others. Mucus, we're actually going to start from the bottom up. This is about lubrication and protection. This helps us to chew our food, moistens the food that we take into our mouth. It's kind of the watery variety of mucus, not super thick, adding or allowing us to chew up our food and kind of swirl it around in there. It also provides a solution that will contain the water-soluble enzymes called salivary amylase. Amylase breaks down starch. Another name for starch is called amylose, which is where it gets its name. If given enough time, it will break down those starch all the way to monosaccharides. In other words, glucose. Okay, that's the breakdown product of starch. Usually we don't hold things in our mouth long enough. It would actually take quite a long time before it becomes all glucose, but you can try this fun experiment if you just go home and get a soda cracker, chew it up really, really good, and just hold it there in your mouth for 2 to 3 minutes without swallowing, and you're going to start to notice that the cracker becomes sweet, or begins to taste sweet in your mouth, and that's due to the fact that salivary amylase is releasing glucose. We also have an enzyme in our saliva produced by the salivary glands, different cells. Sarah's cells, you don't need to worry about that name, but it's produced by different cells in the salivary gland, and this is called lingual lipase. Lingual lipase breaks down triglycerides to produce fatty acids and glycerol. Now, the thing is the amount of digestion in the mouth is very, very minimal digestion. So lingual lipase is actually less about digesting fats and a lot more about it actually digesting just enough for us to help stimulate or recognize taste. So we can actually taste the fat within the cells, within our food. It's hard to describe what this is like. People talk about a mouthfeel, but you can tell when a substance has that richness to it that is associated to fat. It's not like it has a specific taste like salt. It's just kind of a richness that we can detect. And we detect it because of lingual lipase. Moving on to the stomach, we have a number of very important secretions here. And again, I'm going to start from the bottom. First things first, we need to secrete a nice and thick and protective layer of mucus in our stomach. And it's going to coat our stomach lining. It is alkaline, which means it will buffer. Okay, it contains the very important buffer sodium bicarbonate, and that ability to buffer is actually quite important. If we look at the pH of the stomach, the pH is 2, which is very acidic, but if we look at the pH in the close to the cells of the stomach, the pH is usually at about 7. Cells can handle 7 a lot better than they can handle 2. The acid would cause serious harm to our stomach lining, and we actually know what happens when that happens. Without the protection of the mucus and the alkaline buffer there, we end up with ulcers. These are often caused by bacteria called H. pylori, but the effect or the reason we get the ulcers is because H. pylori disrupts the buffering system, causing our stomach to digest giant holes in the stomach lining. This is a process known as autodigestion, when we begin to digest our own flesh. Obviously not desirable. In addition to the alkaline mucus, we have to have something that's going to bring our pH to 2, and we do in fact, and that is our hydrochloric acid produced in the digestive system by cells called parietal cells. These parietal cells are located in what is known as gastric pits. If you look over here on the left, you can see a gastric pit, or a diagram of the gastric pit. The mucus secreting cells are located up here. These are the mucus neck cells. Okay, and that's going to fill this whole region up with mucus to protect it. That's a good thing. We need that. Okay, but then we also have our parietal cells, which you can see shown here in brown, and those parietal cells are going to create acid, which is going to then be pumped into the lumen of the stomach. if we look at these other cells shown in pink, these cells are actually called, I guess we can stick with pink. These cells are called chief cells. And the chief cells produce an enzyme called pepsin. They actually don't make pepsin. They make what is known as a zymogen. A zymogen is an inactive. Let me write that down. Zymogens. You'll hear me use this word a lot. Zymogen, sometimes called a proenzyme. The terms are synonymous. A zymogen is a precursor that is inactive. It's an inactive precursor. In the case of pepsin, the protein is actually longer than it should be. And so that actually keeps the pepsin from being, or pepsinogen from being active. But once pepsinogen gets into the lumen of the stomach, it is activated by hydrochloric acid and other pepsin. And so we end up getting that ready to go and ready to digest substances, but it's in the lumen of the stomach where it away from the gastric pit so that it also doesn't cause autodigestion. So zymogens are a protection against autodigestion too. Move on to the intestines. Here we've got a lot of very interesting things happening. Here's another beautiful slide, and you can see some really just gorgeous goblet cells. Goblet cells secrete the mucus. And once again, it's going to be an alkaline mucus. The alkaline is very important. Most of the enzymes in the intestines work best around pH 7 to 8-ish. And you already know, because I just told you on the previous slide, that the pH of the stomach is 2. And so as the mixture in the stomach, we call it chyme. Let me jot that down. The stomach churns and churns the food that you've eaten and mixes it with hydrochloric acid and pepsin and water, lots of water, as it mixes and churns and squishes and mixes and churns and squishes and digests and all of that. It eventually creates this thick, soupy-like substance. That's what we call chyme. And then that chyme enters into the small intestines. The duodenum, the first portion of the small intestines, is where it's going to enter. It carries with it the acid, and that acid would digest the intestines if we don't neutralize it. And so fortunately, our alkaline buffer contains sodium bicarbonate. As we look at some other secretions, we're also going to see sodium bicarbonate being dumped in bulk into the intestinal lumen by the intestinal epithelium and also the pancreas. That'll be shown to you on the next slide. Very, very important. Now, the intestines can also secrete other things such as enzymes that function in digestion. But the bulk of our digestive enzymes actually come from the pancreas. Hugely important this pancreas is. This is why pancreatic cancer becomes fatal so quickly. The pancreas is definitely a vital organ. Pancreatitis is an inflammation of the pancreas, and it too can become quickly fatal. If you take a look at the pancreas over here, we have the endocrine function discussed on the picture that I just circled. Alpha and beta cells, we'll talk about those in the endocrine system. The hormones are insulin and glucagon. The rest of these are the hormones, the rest of the pancreatic secretions are going to be produced by something called ancillary cells or ancinar cells. You don't need to know that name, but just be aware that those surround the pancreatic duct and begin to empty their secretions in the pancreas. A ton of sodium bicarbonate is dumped into the intestines from the pancreas to neutralize that stomach acid. Again, we're dealing with pH 2, and we need to very, very rapidly neutralize it and bring the pH up to pH 7 or 8 so that the enzymes function correctly, so that we don't autodigest our intestines. Once we get up to pH 8, pepsin is neutralized. It no longer functions. Pancreatic amylase is produced by the pancreas, and this is very similar. In fact, it is the same as salivary amylase. Some minor differences in a sequence of amino acids, very, very minor. The function is the same, and the function is, of course, to digest starch, amylose, and produce monosaccharides, which would include glucose and fructose, among other ones. Pancreatic lipase is produced, again, by that acinar cells, and pancreatic lipase, its job is to digest triglycerides, to produce fatty acids and glycerol. Cholesterol. Cholesterol in our diet is not digested. Cholesterol is absorbed as cholesterol. It remains as cholesterol as it circulates through the bloodstream. It is used to produce hormones such as testosterone and estrogen. Pancreatic lipase alone cannot do the job. It needs to have a coenzyme called colipase. And we'll talk a little bit more about what colipase does to understand how it all works. We also need to take a look at bile. Bile is going to help the pancreatic lipase do its job. Continuing with important pancreatic secretions, we have proteases being released from the pancreas. A protease is anything that digests proteins. There's a number of different names. Pepsin is a protease, although it is neutralized at pH 7, so it's no longer effective in the small intestines. The other proteases have names like trypsin, chymotrypsin, carboxypeptidase, aminopeptidase, and there's others. There's quite a handful of others. All of these digest proteins. They are all produced in the pancreas as zymogens, and again, this is the inactive precursor form of these proteins, and the reason is, of course, to protect the pancreas from autodigestion. They are activated in the small intestines. Their job is to digest the proteins and the peptides and produce amino acids. Let's go ahead and take a look at bile here. Bile is not an enzyme. I can't emphasize that enough. It is an emulsifier. And what an emulsifier does, we actually have emulsifiers in everyday life. Any of our soaps, our dish soaps or body soaps, etc., they're all emulsifiers. And they all work under a basic principle. They usually have some sort of hydrophilic head and some sort of hydrophobic tail. Now, hopefully that looks familiar to you. You should be saying, we've seen that before. Where have we seen that? Phospholipids are like that. Okay, phospholipids are like that. But in soaps or emulsifiers, what happens is as they encounter a drop of fat, they're going to surround that fat. So let's say here's our little fat droplet. The circle is going to represent my hydrophilic head, and the tail is my hydrophobic tail. And they surround that fat and they protect it from the water. Remember, the fat is going to be hydrophobic, and a single tiny droplet of fat is going to find other fat to hang out with. So if you take a whole bunch of little tiny droplets of fat, they're going to merge into one large droplet of fat to minimize the amount of surface area that is exposed to the water. And that's actually not great when it comes to digestion, because that means less surface area for lipase to access. So the bile salts act as a soap to stabilize these tiny little droplets, and we can take a look at that closer over here. So if you imagine that these are very small droplets of fat, let's zoom in over here. Bile acts as a soap. The green circles here are the hydrophilic tail, and it's facing the water. So the water is out here. Okay. And then you've got your hydrophobic tails that are in here, and notice how they're interacting with the fat. And so they bind to the fat droplets and they stabilize the fat droplets and make it so that those small droplets are now stabilized in water. And this increases the surface area then, so that we can now digest that fat more easily. Fat digestion depends on two molecules, as I've mentioned, the colipase and the lipase. The problem with lipase is when the bile salts are in the way, like so, lipase can't really reach them. So colipase helps out. It's not an enzyme by itself, but it helps out by pushing the bile out of the way, providing access to the fat for colipase. And so it'll just kind of spread this out, produces or gives access for this lipase to come in and digest the fats. By the way, the molecules that have a dual nature, such as soap, phospholipids, etc., molecules that have a hydrophilic head and a hydrophobic tail, we refer to these as amphipathic molecules, amphipathic. And that simply means two paths, more than one path. And so you can have those types of molecules, and they make great emulsifiers, great soapy layers that can be used to increase the surface area and therefore increase the digestion of fats in this case. Or if you're washing dishes and the dishes are greasy, they can help the grease bind on to our little hydrophobic tail, and then the hydrophilic head will interact with the water and it will wash down the drain. Absorption. We're going to get into the details of absorption in the digestion lecture at the end of the semester. For right now, I just briefly want to introduce this idea to you that we are going to be getting molecules passing through the epithelial barriers, and those molecules need to be small. So our starch has to be broken down. So if we have our starch, we're going to break it down into trisaccharides, disaccharides, sucrose is the disaccharide, and then eventually into glucose and fructose, so that we can absorb this glucose and this fructose transcellularly into the bloodstream. And that would be through a series of transporters. I won't get the specifics for you, but there would be a glucose transporter right here, for example, and a fructose transporter right here, for example. And so we would bring the molecules into the cell, and we would export them out of the cell. Concentration gradient would matter quite a bit. So if we eat a diet high in carbohydrates, we're going to have a high concentration of glucose in here, maybe a high concentration of fructose if we eat some fruit, and that will help to drive the sugars into the cell. So that would allow us to move sugars in by facilitated diffusion. Whereas on the other side of the cell, usually we've got a low concentration inside the cell because glucose tends to be used for cellular respiration, or in the case of the digestive system, not used for cellular respiration in large quantities, but instead maintained low because we continue to pump it out. And so on this other side, we would have a pump like so, or a pump for the fructose like so. And in this case, we would actually be using active transport because we're going to be pumping it from low to high-ish. Usually blood glucose in the blood is maintained at about 100 milligrams per deciliter. Okay, and that's certainly higher than what is in the cell. And so now we're going against our concentration gradient, so this is active transport. Proteins and peptides are way too big, and so we're going to transport those by converting them into amino acids. We do have a mechanism that allows us to transport and even peptides that are up to 13 amino acids long. That's actually something that viruses have a tendency to exploit because they can get in bigger peptides that way. Fatty acids are quite interesting in that, triglycerols are quite interesting in that we have to break them down into glycerol and fatty acid, transport them into the cells, glycerol and free fatty acids, and then we actually reassemble them and export them through exocytosis. Sorry, that's all scribbly and messy on the other side. And so this just kind of walks you through that process here. Here I talk about the fatty acids in the apical membrane. Up here I'm talking about the difference between the proteins on the apical membranes and the basolateral membranes, and you're going to want to understand that because it certainly becomes important as we go through just our understanding of physiology and how we move things and how we maintain homeostasis. Large intestines, just briefly, primarily involved in the absorption of water, some vitamins, not much else. Bacteria in the large intestines are going to digest the leftover material, and with the help of the bacteria as well as the removal of water, we're going to end up forming the feces that'll be excreted via defecation. And so that's basically the overview of digestion. Now, another piece of information you're going to need for this lab is succinate dehydrogenase activity. Succinate dehydrogenase, also known as succinic acid dehydrogenase, is an enzyme found in the citric acid cycle. It's located in the mitochondria. Its job is to convert the sugar, succinate, also known as succinic acid, into the sugar fumarate, or fumaric acid. This chemical reaction will produce a molecule of FADH2. This is a redox reaction. Okay, this is a redox reaction, and remember that it's occurring in the matrix of the mitochondria. When we look at the redox reaction, succinic acid is being reduced to become succinic acid is being oxidized to become fumaric acid. So fumaric acid is the oxidized state. And FADH2 right here is the reduced molecule. Okay, is the reduced molecule, and so that's actually pretty important to understand. This would also become reduced, so it's reduced to the oxidized state. It's oxidized to the oxidized state. FADH2 is reduced, and that's essentially our A meets B reaction that you saw previously. In this case, B is FAD, and it becomes FADH, and A is succinic acid, and it's going to become fumarate. All right. Another reaction not shown here is oxygen. Oxygen will react with FADH to produce FAD and water. You need to remember this because at one point in our experiment, we're going to be asked to shake the test tube, and the reason we shake the test tube, or what happens when we shake the test tube, is it introduces oxygen. So keep that in mind when you are writing your data up and analyzing your data and trying to figure things out. That's actually an important little detail there. One other thing I want to mention, and for this I actually think I'm going to go over here and open up a new slide really quickly, is what happens when FADH encounters a molecule that is usually not found in the cell. When FADH encounters this molecule not found in the cell, it is converted back into FAD, and the molecule that we're not finding in the cell, but we are going to add in the lab, is methylene blue. Okay, methylene blue. And methylene blue picks up those extra hydrogens, so it becomes reduced. And the interesting thing is that when it's reduced, it actually changes colors. Instead of being blue, it's now clear. Okay, instead of being blue, it's clear. And so that actually is going to be used as a tool for us in lab. We're going to use this reaction, this redox reaction, As more FADH2 is produced, more methylene blue is reduced into a clear form. And so our reaction is going to start out being blue, and it's going to proceed until it's clear. And each time we do this, we introduce FADH. Interestingly enough, oxygen will encounter our reduced form of methylene blue and turn it back into our oxidized form, which happens to be blue. I ran out of room, so we're just kind of scribbling that in. And oxygen restores the color, so keep that in mind when you're shaking your tube. All right, store that away in your memory, because once you do this experiment, it will become clear. All right, what I want to do next is not walk you through the experiments. We don't have a lot of time today, so we need to get you in, get you started, get you working on these experiments. You need to make sure you read this in advance. You pay careful attention to how long you have to let things sit if this is going to be successful. There's not a lot of room for mistakes today, so hopefully you plan ahead, you think it through, you're ready, and you get going. We're going to divide you up into groups. So most tables have 4 people in their group, in their table. We're going to split you into 2. Half of the group is going to do one half of the experiments, the other half is going to do the other half of the experiments, and then you're going to share your data. Make sure you make note of who you're getting the data from, you know, so that you are giving credit where credit is due. That's how we do things in science. We're ethical about it. We say, okay, this data was from so-and-so, and then that person would get, you know, if we were in theory going to publish the data, we would give that person an authorship on the lab, or on the publication. Okay, experiment A, let's look at that fairly quickly. We're going to look at a total of 7 test tubes. We're using a lot of test tubes in this experiment, a lot of test tubes. Make sure you give yourself a lot of time to clean up. I'm going to try and get you guys to stop about 30 minutes early, or at least 20 minutes early, because you're going to have a lot of test tubes to wash and clean. So experiment A is actually about getting you familiar with what we call the indicators. Indicators are things that change color or that produce something that we can see with our eyes or detect with our senses. It could be an odor, it could be a precipitate, it could be a color change, that type of a thing. Those would all be considered indicators. And they let us know that a chemical reaction has occurred. The first indicator that I want you to familiar with is iodine. And so the first 3 tubes you're going to set up. You're going to put starch into one tube, glucose into the other tube, and you're going to put water into the 3rd tube. Okay, there's nothing specific about these colors. I'm not really color coding. And then in each of these you're going to put iodine. Now iodine is kind of an amber color. In the book it will often call it Lugel's iodine, but in the baskets that you have it's just straight iodine. It's the exact same stuff. So don't stress out and be like, where's the Lugel's iodine? It's in your basket, even though it's just called iodine. Usually iodine is kind of an amber or a brown color, and then we're going to see a color change when it interacts with either starch or glucose, and you get to look at that and learn what color change that is. You're going to set up another 2 tubes, and this time you're going to add glucose and you're going to add water, and the glucose and the water will then you'll add Benedict's to it, and you're going to see if you can get a reaction. Benedict's has to be boiled, so you're going to cover the tubes and you're going to place them into a beaker of boiling water. Please make sure you actually put them into your beaker. So here's my beaker. And you're going to wrap the tubes up, and you're going to set the tubes into the water like so. If you have it wrapped up, if it tips a little bit, it should be okay. You're going to wrap it up with a substance called parafilm, and there should be parafilm on your desk or in your baskets. You're going to boil it for about 2 minutes, 2 to 3 minutes. 3 minutes to be safe, until you get a color change, and then you're going to observe what that color change is. Then the last is not so much a control as it is an experiment. You're going to look at the effects of amylase and how it affects starch, and what happens when you put amylase in a solution with starch. Now, you should already know what the chemical reaction is. I've already told you from your digestive enzymes lecture just a few minutes ago that amylase digests amylose, also known as starch, and it digests amylose and produces from amylose monosaccharides, or sugars. Okay, you should know that. And so you can probably make a reasonably good hypothesis. If I put starch in water, should I get any kind of digestion of the starch? Will starch just spontaneously break down? But if I put starch in amylase, what is that starch going to break down into? You're going to put these tubes together and you're going to incubate them in a water bath at 37 degrees Celsius for 40 minutes. You'll find the water baths in the back. There's 2 of them. Make sure you label your tubes. Always label. Always. You probably should include your initials. Okay? Because otherwise you might get your experiment mixed up with somebody else. Once you've incubated these for 40 minutes, again, pay attention to timing here, 40 minutes. So it's going to take you a few minutes to get everything set up. Then you're going to do it for 40 minutes in the water bath. Once you've done that, you're going to take these out of the water bath, shake them gently, and observe their color. And then you're going to add your Benedicts to them. And they're going to boil them in water for 2 to 3 minutes. 2 to 3 minutes. And from there, you're going to pay attention to color change. Do you get a color change in the tube that had water in it? Do you get a color change in the tube that had amylase in it? What is the color change and what does it mean? What is the significance of that? Once you have that information, you're going to move on to this next experiment. Now this one gets pretty complicated. This is experiment B. You're going to gather 8 test tubes. You're actually going to use a lot more than 8. But you're going to take 8 of the test tubes that you are going to use, and you're going to put starch in the test tubes. That's it. That's all you put into it, the starch. Now you'll notice these pictographs, that's what I'm going to call them, just picture images to give you a visual image of what you're doing. You're noticing I'm not including any kind of concentration or volume or anything. You need to check your lab book for that. This is just a quick picture, and you're welcome to print this out and write all over it and whatever you want to do. But this is a quick picture to tell you what's going into the test tubes. So all of these contain starch, and you're going to set them aside. And they're just going to sit there, and you're not going to do anything with them until you're ready for, until you've done the other steps. Now you're going to gather another 8 test tubes. Again, you want to make sure you label them. Label them with your initials. Label them with numbers. Okay? And in one of these test tubes, you're going to put just plain old water, nothing fancy, just water. In one of these test tubes, you're going to put a little bit of water, so 2 milliliters. Another test tube, 1 milliliter, and the third test tube, no water at all. Then you're going to put amylase in it. 1 milliliter of amylase, 2 milliliters of amylase, 3 milliliters of amylase. In this particular figure, I did include the volume because what you're really doing, what you're really looking at, is concentration. This one is full concentration. This is the maximum concentration that you have. And it's whatever the bottle happens to contain, okay, whatever that concentration is, because you're putting it in full strength, right? Full strength. But this one, you've diluted it, so now that's not full strength. That's essentially 2/3 of full strength. And this one is 1/3 of full strength. And this one has nothing in it. So it's 0. Okay, and that's essentially what your concentrations look for. In the lab book, it's going to ask you about concentrations. I actually don't expect you to calculate it out. You can if you want to. It should say on the bottle what the concentration of amylase is. but I'm actually okay if you essentially use these to represent your concentration values. Okay, full strength, 2/3, 1/3. This is your negative control. I shouldn't have told you that. You should get in the habit of identifying your negative controls and your positive controls when available, so that you can practice this. Importantly, you need to control you need to compare the pH for this one and this one. You're not going to change the pH in any way. You're just going to record it. pH of the distilled water, I think it's usually at about 5, but you're going to find out. So you're going to stick a pH strip in there and find out what the pH is. Or better yet, get a pipette and drip the water onto the pH strip so you don't contaminate your sample. Same thing with the 3 milliliters. It should be roughly equal in terms of pH, but it might not be. Okay, it might not be. Now we're going to move on to some more tubes. Now we've got 5, 6, and 7. Remember, you still have the 8 tubes containing starch set aside. The next tubes, we're going to put 3 milliliters amylase. So notice you're putting in full strength now. Full concentration, 3 milliliters of amylase into each of these tubes. You're going to mix it. One of these, number 5, goes on ice. One of these, number 7, you're going to take this, make sure you cover it. You're going to put it into a beaker full of boiling water, and you're going to let it sit there for 10 minutes. Okay. Now you can probably guess, what do you think boiling water is going to do to that enzyme, amylase? Do you think amylase loves to be boiled? You take a guess, you make that decision. The last one simply stays at room temperature. You're just going to leave it on the desk. Depends on what the temperature of the room is. Typically it's about 22 degrees Celsius, although it's been running a little hot lately, so it might be 20. I think the last time we were in there was like 25 degrees Celsius. Somewhere along there, you'll make a note of the temperature. There will be thermometers available. So now we've got 1, 2, 3, 4 all done, 5, 6, 7 done. And once you are incubating these for 10 minutes, you're going to proceed with the experiment once you're done with those. Tube 8, full strength amylase and HCl. Now, you needed enough HCl to make your pH less than 2. We're going for a pH, our goal is a pH of 1 to 2. You will need probably 10 drops or more. You will check your pH frequently to see where you're at. So you're going to add some drops of HCl, check, add some more drops of HCl, check, add some more drops of HCl, and so forth until you get to a pH of 2. It's very important that you actually get down to that pH of 2. All right, we've got all of our test tubes prepared. You have placed number 5 and number 7 on ice. Okay. Now 10 minutes have passed. Go ahead and retrieve number 5 and number 7. Put them in your beaker. Add add iodine to each of these test tubes. Notice my pH. This is going to depend on what you measured. So if you measured 5, you're going to record that. If it's 7, record that. Okay. Each of these get some iodines. This is a little bit of a reversal from your manual. I think it's easier to add the iodine before you add the starch, but if you don't want to do it that way, you can still do it the way your manual says. Regardless, all 8 tubes get iodine. And now remember those starch tubes that you set aside, tubes 1 through 8 that contain nothing but starch. You are now very quickly going to dump each of these tubes, and it really doesn't matter which tube goes where. Okay, but starch goes into each of these tubes. Okay, quickly, as fast as you can. You want to try to get the starch in there as close to the same time as you want, because as soon as the starch enters into those test tubes, it is now go time. This is when your time 0 is. Make a note. Make a note. Mix them up well. Cover them up with parafilm. Transfer 1, 2, 3, 4, 7, and 8 to the water bath that's at 37 degrees. Put your number 5 back on ice. Okay, put your number 6 back on the desk. Remember, 7 is the one that you boiled. but you're not going to put it back in the boiling water. Just put it in a 37-degree water bath. Number 8 is the one that you lowered the pH. So this is the one that's at pH 2, hopefully, if you did things correctly. 60 minutes later, you're going to retrieve your test tubes. You're going to shake them. You're going to make note of the color. You are going to use plus symbols to represent the extent of digestion of starch. Iodine. Remember, iodine changes color in the presence of starch, and as that color change is lost, then that would represent an absence of starch. So if iodine turns blue, for example, and then starch is digested, so blue indicates starch is present, but the blue is lost, well, what does that mean? It means your starch has been digested, and that's essentially what you're going to keep in mind to interpret the results of this experiment. When the blue color turns into white or clear, it does so because that amylase broke down all of the starch. Iodine interacts or reacts with starch. It does not react with sugar. Then you're going to take a sample of each of these tubes. 2 milliliters. Transfer them into brand new, clean, fancy tubes. And you are going to then add Benedict's solution. You're going to cover them all up, and you are going to boil them. Benedict's solution interacts or reacts with sugar, specifically monosaccharides. It will also react with lactose. Keep that in mind. The sugars, again, you know, amylase. Remember this. Amylase takes starch and make sugar. Okay, so if we go back up here and starch becoming sugar means that our blue color disappears. Well, what happens when starch becomes sugar down here? Well, now instead of our blue color disappearing when we boil that Benedict's, it's going to go from blue to orange as more and more sugar is being produced. And so once again, you're going to use plus, 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 plus to indicate the extent of the sugar production. 4 pluses means you're looking at a nice brick red color, very vibrant. 3 pluses, it's probably going to be kind of more of a dilute orange or an orange yellow. 2 pluses is usually classified kind of as a brownish color, maybe a greenish color, or a yellow green, and 1 plus is just a hint of brown in it. Remember, these are subjectives. These are my colors for them. You might decide that it doesn't really look brown at all. It looks a different color, but the idea is essentially intensity, with the brightest, most vibrant color being the one that has the most intense production of sugar. And that's experiment B. I want you to make note of, you've got 8 tubes there, 8 tubes here, and 8 tubes of starch. So you have quite a few of them that you are going to be using. So lots of tubes. Make sure you plan ahead to clean those up. The Benedict solution can be a little bit difficult to clean. You're going to use dish soap to clean those test tubes, but if not, if it still is orange, then there's a paste in the back. This should be by each of the sink. You can put some of that paste onto a bottle brush. Those should also be back by the end of the sink, and use it to scrub out your test tube. Do the best you can with it. Okay, remember, I'm going to be checking to make sure everybody's doing a good job cleaning up the lab. Moving on to experiment C. Lots of tips and tricks for this one. I want you to pay attention. First, you're going to mix up salad oil and water, shake really well. Salad oil, water, and bile salts, shake really well. Take a drop of each, and transfer them to a test tube. You're going to have 2 slides. I'm sorry, not a test tube, 2 microscope slide. You're going to have 2 slides. One slide is going to contain your salad oil and water. One will contain your salad oil and bile salts, and of course water. Okay. You're going to place both of these slides on a microscope. My strong recommendation, your lab manual does not tell you to do this, but my strong recommendation is to get out 2 microscopes, especially since we're dividing up the lab. You'll have enough microscopes to do this. Set both of the microscopes up on your desk, and in one microscope put a little sticky note on it if you can find one, or some kind of little piece of paper that you just slide under it so you can keep track of which slide's which. One microscope you're going to place the salad oil and water slide on. The other microscope you're going to place the bile and water slide on. I'm sorry, the bile and oil and water slide on. And you are going to focus both microscopes, and you're going to observe at 40x, and you're going to draw your observations. This works much, much better than trying to use one microscope. With one microscope, you have to put slide 1 on, observe it, take slide 1 off, put slide 2 on, observe it, take slide 2 off. And between each of those, you might lose focus. So by doing 2 microscopes, you can actually leave everything in place. All you have to do is turn off the light, and then when you come back 30 minutes later, you turn the light back on. You might have to do some fine adjustments with the focus, but not significantly. You should still be in about the right plane of focus. And then you're going to do your drawings, turn the light off, and repeat. Make sure you turn the light off. If you leave this on, it will cook. It will cook your water away, and your experiment is over and it's done. Because you're not going to see anything if there's no water for the oil to float around in. All right. You should see over time changes, a difference between what you see in the microscope with the salad oil and the water versus the bile salts. Remember that bile salts are an emulsifier and they stabilize smaller droplets. Experiment D. Okay, and this one, we're going to also deviate from your lab manual a little bit. I want you to make sure that when you're done with these solutions that you add enough sodium hydroxide to each of the test tubes to make it a pH 8. It should be a bluish or purplish color. Okay, it should be on the blue side of the spectrum. All right, if it's not, then you're not going to be able to notice the difference in the colors. Once you get it on the blue side of the spectrum, so one of them is going to contain evaporate milk and litmus solution. This is actually in a bottle. Most of you know about litmus paper. That's your pH strips that you put in here. Okay, you dip those in, and we are going to use litmus paper. In fact, when it says check the pH with paper in your kit, this is what it's talking about. But the litmus solution actually is in a little bottle, a little squeeze bottle with a little cap on it, and you're going to use that to squeeze in your litmus solution, and you're going to put it in your evaporated milk. Please make sure you use the evaporated milk that contains fat. If you use the non-fat, your experiment is useless. Pancreatin contains, let's talk about what's in pancreatin. Pancreatin is basically, you take the pancreas of a pig or a sheep or a mouse or whatever you're going to use, you grind it up into this blender, and then you extract through a series of different steps, centrifugation and so forth, you extract the liquid portion. It's going to contain all of your enzymes, but it also will contain lipase. It's the lipase that we are interested in this experiment. Lipase digests fat. If you put non-fat milk into your experiment, there is nothing for the lipase to work on, and your experiment will not work. So make sure you're using there's 2 types of milk. One of them is non-fat, one of them is full fat. Evaporated milk, lots of fat, that's the one you're going to use. Goes in the water bath for 60 minutes. Once you've got it all set up, please, again, I cannot emphasize this enough, it needs to start out basic. If it's not basic, you're not going to see a change, or you're not going to be able to detect a change. Okay, experiment E. We're actually going to skip this experiment. It tends not to work well anyway, and we're short on time. So basic strategy, I want you to know how the experiment is done. So at least the step-by-step process. This is what you would have done if you had done this experiment. And I'm going to walk you through the results of what you should get, your expected results. Just because they're expected doesn't mean it always works. And as historically, as in my lab, it's very common to get slightly different results. One of the biggest problems is we actually use an acidic soap when we wash the test tubes. And so it's a pretty good idea always to rinse out your glassware anyway, but it's also very common for the acid in the test tubes to activate the pepsin. And so you end up getting a digestion when you shouldn't. Regardless, I want you to notice how long this incubation is. That's one of the reasons I'm taking it out of the equation. But what you would do is you would fill this up. Each of these beakers would be filled up with your solution. Each solution is different. For number 1, the solution is a pH 7 buffer. For number 2, the solution contains buffer and the enzyme pepsin, isolated. 3 contains the buffer plus HCl. 4 contains pepsin plus HCl, pepsin plus NaOH. NaOH is sodium hydroxide and it is a base. HCl is an acid. And here's the buffer plus the NaOH. So we have these 6 different conditions that we're looking at. Based on what we know about pepsin, it's most active at pH 2, and so you would expect the one I just put a star on to be the most active. Now you're going to take a piece of X-ray film. It's a black little square like so. And it's black because it's been exposed. Okay, it's been developed. During the developmental process, what you actually do is you, well, never mind, I don't want to get into the chemistry of it. But the effect is, or what's going on is you're actually holding proteins onto a plastic strip. And it's the proteins that give you that black color. And then on top of the proteins is a silver coating that is there, and so you get this black strip of film if it's been exposed to the light and then developed. You're going to put one of these strips in each of these little black pieces of film. The strips are cut. They're actually slightly different sizes. Ideally they would all be exactly the same size, but let's be real. Life being what it is, we're very lucky to have a wonderful team of lab techs preparing this for us, for the hundreds of students that do these labs. So, you know, we can forgive minor differences in sizes here. And so we're going to put these black pieces of film into the beakers. 90 minutes. Then we're going to take the films out with a pair of tweezers and we're going to very gently run it under water. What we should see is as we take this film out, this one should be remaining pretty much completely black. 7, pH 7. This one should be made mostly black. There might be some minor feathering at the edges that contain a slight clear, we'll do this one, a very slight translucent color at the edges, maybe. The next one should again be mostly black. HCl can denature proteins, don't forget that, so sometimes you'll get a slight feathering again, and actually in this one maybe even just a little bit more feathering than is you might otherwise expect because of the ability of HCl to emulsify, so we might get a more partial or mottled look with the maybe something like that. Okay. And then pepsin with the HCl, this is ideal conditions, so hopefully if everything works the way it's supposed to, the entire thing has become this translucent blue. Pepsin with NaOH, no, it does not work under basic conditions, and so that should be pretty much no change, all black piece of film. And then pepsin doesn't exist in this one, and so again, all black piece of film. You might actually get a little feathering on this one too, because again, NaOH, acids and base both will denature proteins, and so if you denature the proteins you might lose some of the silver from it, but generally not. So that's essentially what you should see if the experiment works perfectly. So I want you to record those in the results as if you had done this experiment. Just make a note that this was a thought exercise, not an actual experiment. 4, we're also going to skip because again, for time, I need to reduce your workload if we're going to get through this lab. And so what we're going to do here, what logically, or not logically, what I want to do is walk you through the process here. You're going to get another test tube. You're going to put an enzyme in it called renin. Now what renin does is, in milk you have these soluble proteins. So if we were to look at a glass of milk, okay, what you're going to see, if you looked at that, if you could magnify it enough, you would see, of course, droplets of fat, although in this case we're actually using non-fat milk. That's important to simplify the experiment. But you'd also see these soluble proteins. Now, milk is actually called a colloid. And what that means is the proteins aren't actually dissolved in the milk per se, but they are in suspension. They're small proteins, very small. But when you add renin to that, what renin does is it breaks down those proteins and it causes them to essentially become insoluble, and they will fall out of suspension and they will make a solid mass at the bottom. And so we end up with this thick substance. You might have heard of curd. That's what this stuff is. The liquid portion on the top is whey. Little Miss Muffet sat on the tuffet eating her curds and whey. That's what it's talking about. Add the renin and you end up getting this curd. Sometimes renin is also called rennet. Can't remember if it's 2 T's or 1. Regardless, it's the same molecule or the same enzyme. Don't confuse renin with 2 N's with renin in the renal system. It's not the same thing. Okay, and we'll talk about renin when we get to the urinary system. All right, so once we do this, what you do is that you actually take a sip off of that whey. You actually collect all of the whey if you can. So all of this stuff here transferred into a new beaker. Here's my new beaker. Transfer that whey into here. So now we've got our little whey here. And then you're going to add your Benedict solution to it. And that Benedict solution will, hopefully you're doing about an equal volume, approximately as close as you can get. Boil that. And what you should see, this is the big question mark, what you should see is that this should change colors. And you should indeed get a nice orange color after you've boiled that for 2 minutes. Here's an example of what it would look like. So this is my negative control. This contains only water. Get in the practice of recognizing what your negative and positive controls are. There is no positive control here. And this would be my whey with Benedict's after I've boiled it for 30 minutes. Sorry, 3. Please don't boil it for 30 minutes. Way too long. 3 minutes is enough. Usually 2 minutes is enough. As soon as you get a color change, you can stop. That color changing indicates that, remember Benedict's, what does it do? Well, it responds to sugars that can be reduced. So it simply tells you the whey contains sugar. Does it tell you exactly what sugar? No, but it's probably glucose. Milk contains lactose, right? Lactose. And lactose is a sugar, it's a disaccharide. And one of the saccharides in lactose is glucose, in fact, and so that's what the Benedict is likely reacting to. Experiment G. I think, in fact, I'm just going to make the decision. As your instructor, I will do this. And you will send somebody up to my desk periodically to check on the progress of this. Again, I'm just trying to lighten your load. We've got a lot to get through on Wednesday to get this done. This is like one of the worst labs we can possibly try to cram into one lab. But we're going to do it. We're going to succeed. So essentially, experiment G. Here's what I'm going to do. I'm going to take a chunk of hamburger. Yummy. It's actually going to be ground up hamburger shake. It looks really gross and delicious. So I'm going to put a chunk of hamburger into each of these test tubes. 1, 2, 3, and I'm going to try to do approximately the same amount of hamburger in each. But notice this one does not have any hamburger, so there's no hamburger in that one. Then I'm going to add succinic acid and methylene blue into my solution. Methylene blue is, well, blue. But with the hamburger meat, it's going to turn this really gross, sickly color. Okay? Kind of like a greenish color. It just looks really super nasty. And And in this one, it'll look pretty because there's no gross hamburger shake to make it even really ugly. Note that this one contains succinic acid. Okay, hamburger plus succinic acid. The next one contains water. Okay, third one contains malonic acid. Nah, what is that? Malonic acid is an inhibitor. Think back, I showed you the citric acid cycle and the conversion of succinic acid to fumaric acid, or succinate to fumarate, with the aid of the enzyme succinic dehydrogenase. You'll want to flip forward to the citric acid cycle if you need to take another look at that reaction. Malonic acid inhibits succinic acid dehydrogenase. So imagine, if you will, my little Pac-Man dude is my succinic acid hydrogenase. Okay, well, normally what that's going to do is it's going to bind to my substrate, which would be succinate, and it's going to convert it into fumarate. That's the normal thing. So that would be my normal reaction. But when malonic acid is in the picture, malonic acid, let's draw it as a, what do I want to do? We'll stick with brown. Okay, it's a competitive inhibitor. It's going to get in the way, and so when it's in the way, my succinate can't bind. And so if I can't get my succinate to bind, I'm not going to produce fumarate. The hamburger in this reaction is my source. Okay, hamburger is essentially skeletal muscle and fat and blood vessels and tendons and all sorts of stuff ground up, right? Yummy. And the skeletal muscle contains a ton of mitochondria. So my hamburger is a source of mitochondria, and my mitochondria is a source of succinic of my enzyme, succinic acid dehydrogenase. You can buy succinic acid dehydrogenase on the market, but it's pretty expensive. Hamburger meat is really cheap, comparatively speaking, especially in the quantities we use. We're not using that much. And so we use the hamburger. Don't forget that. The hamburger is the source of the succinic acid dehydrogenase. So in each of these cases where you see a hamburger, you can think, oh, I just added enzyme. Oh, look, there's some enzyme. Oh, look, there is no enzyme. No enzyme. So what does that make this? It makes this my negative control. Okay, no enzyme, negative control. All right, don't forget with this particular one that we're also going to be using that methylene blue. And again, the reason for the methylene blue is this. Look, let me kind of draw it all out for you. And I'm not going to draw chemical reactions. We're just going to draw, I'm just going to write succinate becomes fumarate, and that's with my succinic acid dehydrogenase. Sad. Okay, that's my enzyme. But we're also going to convert in the process. In fact, I'm going to move my enzyme to my other side. Dehydrogen, that doesn't fit. We're going back to sad because that fits. Succinic acid dehydrogenase. But on this side, we're going to have my FAD. Remember, this is reduction oxidation reaction, FADH. Okay, can you see FAD? Can you see FADH? Can you see succinate? Can you see fumarate? No, you can't see them. We're not watching this with a microscope, and even if we were, we don't have microscopes powerful enough to see individual proteins. We just don't. We have to do things like X-ray crystallography and then use microscopes. We can use X-ray diffraction, but we can't see individual proteins. We just don't have that technology yet. So since we can't see this, we need to figure out whether this reaction is working or not. We need to find some way to monitor it. And so the way we monitor that is by putting in our blue, methylene blue. And remember, that's going to become clear. It's still named methylene blue. It's not like I can say, oh, it's methylene clear now. That's silly. That's not its name. But it's the clear form or the reduced form. And in the process of doing that, it's going to actually regenerate this FAD. Okay? And so, can we see this? Well, we're not going to see the individual molecules of methylene blue. No, not at all. But we definitely can see a color change. And so over time, we're going to start to see the blue color fade away. And we're going to begin to get the liquid clearing up and becoming translucent and clear. Don't forget, our good friend oxygen can combine with this, produces water, and turns this back into the blue color of methylene blue. And so we can see that too. So color is all great and wonderful. Things that change color make wonderful indicator dyes. That's a terminology you should be familiar with. Indicator dye. Indicator dye. Not all indicators are dyes. Silver nitrate, for example. That's used as an indicator. What does it do? It doesn't turn anything colors, but it does form a precipitate. So not all indicators are dyes, but we often use dyes as an indicator. All right, hope that makes sense. We will see you in lab on Wednesday. Bye-bye.